Well, good morning, everyone. Good to see you. My name's Paul Kruszka, as James said. First of all, thanks for PacBio for inviting me to talk about some of the really cool work we're doing at GeneDx. I'd also like to thank Joe Devaney. Are you out here somewhere? There, my partner at GeneDx and his team that's here, who helped me with these slides. So I'm a clinician, and I need some help with some of the technical pieces. I've been practicing for almost 3 decades. Still practice at Children's National Medical Center on Fridays. And like James said, I'm Chief Medical Officer at GeneDx. And GeneDx is really it's been a wonderful place to work. I've been there 3 years. We spun out of the NIH. Sherry Bales and John Compton about 25 years ago formed this company. And I've been here 3 years, and it's really it's an interesting place to work. It's certainly on the forefront of genomics. About 8 out of every 10 exomes or genomes that are commercially ordered come through GeneDx, so we've got this incredible database. We have over 600,000 exomes right now in our database, and we're trying to obviously publish and get this data out there, share it with the ecosystem, because there's really a lot more to learn right now about diagnosis and genetic disease. And I use this metaphor on my slide of this person who's had too much to drink, and he's looking for his wallet. This is a common metaphor in genomics. He's looking for his wallet. There's the pointer, and a police officer comes by and says, "Why are you looking here?" And he says, "This is the only place I can see." And we often think in the genomics world as the current technology that we have. So, and so that's really new technologies, expanding this light and trying to find more answers for people is really what it's all about, at least for the diagnostic piece of this. And so we're living in an exciting time right now with new gene therapies, new small molecules. We're really entering this era of treatment as opposed to diagnostic. I feel like my career was in diagnosis, and now it's an exciting time. And there's still a lot to do in diagnosis. If you send your sample into GeneDx, and obviously there's some ascertainment bias, people tend to only send us the sickest of rare diseases. Our diagnostic rate is probably between 20 and 30% roughly. So we're missing 70%, and that's why I decided to talk about what are we missing, and I thought it matched up perfectly when we talk about long-read sequencing. What's the next step? What are we missing? So just out of curiosity, I see Carlos Ferreira in here. I know he's actually the best, the #1 physician I've ever met in my life. I worked with Carlos at NHGRI for a long time, so a lot of respect for him. Besides Carlos, are there any other clinicians here? Just so I'm trying to, I know Dr. Greeley, of course. Yep, I know you. Anyone else besides John and Carlos? And John, you should meet Carlos over there before you leave today. He's an amazing guy. Anyone else? Oh, good. So I can teach you something about clinical genetics then. So, all right. So for my talk, I'm going to talk a little bit about the present state of clinical genomics. Certainly, I have that perspective as Chief Medical Officer of GeneDx. I can kind of tell you the lay of the land, what's going on in North America. I'm going to talk about some collaborations we're doing in long-read sequencing, because that's why we're here. I'll talk a little bit about our workflow, and then a few of case studies that are really interesting. I can show you some things we're doing. And then I want to talk a little bit about a future state, a vision, and potentially the end of the diagnostic odyssey and some work that we're doing. So when you think about genetic technology, there's certainly different stages here, and I love this graph. In the 1970s, we started out with Sanger sequencing. In the early 2000s, we moved to chromosomal microarray. In the 2010s, next generation sequencing, and it's no secret that the next step is really going to be long read sequencing, which is here right now, and this will be the continuum. And it's a really interesting continuum because, you know, many of us have lived through this technology advance, and I use short read sequencing as an example of how to think about long read sequencing and where we're going. Mike Bamshed, who I'll talk about a little bit later, along with Dr. Greeley, John Greeley, in 2010 in Nature Genetics, did the really, which I think is the first genotype-phenotype relationship using short read technology, exome sequencing. They associated Miller syndrome with a gene, and then the paper that really caught more attention, I believe, was the Kabuki syndrome paper, also in 2010, and this was Jay Shandor's group. And up until this point, we weren't finding new genetic syndromes with short read sequencing, even though it had been around for a few years. So this is 2010. The Kabuki paper comes out in 2010. GeneDx finds its first patient with Kabuki syndrome using short read sequencing in 2014, so 4 years later. So this is, I think James talked about technology adaptation, so this is the technology adaptation curve. So 4 years later, our turnaround time for that case was 4 months, and so, but we did it in 2014. Let's fast forward to where are we today, 2024. Short read sequencing is the standard of care. Most guidelines support it. ACMG recently put out guidelines a few years ago making exome genome short read sequencing a first-line approach to a lot of different conditions. But I can tell you there's only 28 out of 50 states that will cover exome for Medicaid. So for children with a rare disease, 28 states. And so we're you know, this is a long curve. The technology curve, adoption curve is long, and it's a really good example of where we are. And certainly, for us, if you're a clinical geneticist, you want long read sequencing now. and I think the talks today will certainly explain this, why this is. So since introduction of short read sequencing, certainly the genotype-phenotype relations have blossomed and increased over the years. It's exciting. And I guess this never gets old. You know, when a new gene or a new condition comes up, I don't know if any of you guys read that recent preprint RNU42. Has anyone seen that paper? It's a preprint. It's a non-coding gene. It's involved in it's part of the spliceosome. And there's a preprint out there. If you email me, I can send it to you. But I believe that maybe 1% of our population at GeneDx has this. So it's just really exciting, the new advances in this curve, and this never really gets old for me. But there are shortcomings of short read sequencing. I think James went through a lot of these. And for the sake of time, I might refer to what James talked about. I think he laid out some of the shortcomings, and certainly we all, especially in the clinical side, but also in the laboratory side, looking for that one test. We always talk about what's the one test. We want to do one test. We don't want to do all these tests, MLPA, Sanger sequencing, gel-based assays like Southern blots for repeat expansions, triple prime PCR, all the karyotypes, all these different types of tests that we do. And we know if you get into the nitty-gritty that short read doesn't cover a lot of these. And if you're, especially from our perspective at a commercial lab, it hurts us not to have that one test. So, you know, the question is long read that final test? And I really believe it is. Here's a preprint that just came out. This is from Hudson Alpha, and I was not an author on this paper. I just saw this paper come through the ecosystem, and I thought this was a really nice preprint. I'm sure it'll go to a top journal because it lays out, it answers that question, what's the diagnostic yield? What do you get from long read sequencing? How much more yield? And that's something I'm always thinking about, because we want to end the diagnostic odyssey. That's the game here. And this was a study, 96 patients who had short read sequencing. 94 of them were genome, 2 were exome. Most of them were neurodevelopmental disorders. So 70% were neurodevelopmental disorders, 20% congenital anomalies, and they report a 7.3% increase in yield from short read. And some of the things they found: copy number variants, inversions, multiple element insertions, low complexity repeat expansions, and a one base pair deletion. So this is the type of evidence certainly that we want to generate as a community and answer these questions. How much more can we get out of long read? And certainly this is going to change. There's new conditions and new diseases and new applications that are always changing. So some of the things we're doing to move the needle forward, we bought a Sequel IIe in 2022, and it's been fun. I came to GeneDx in 2021, so I've been able to watch all of this. And then we bought our first Revio later that year, and we just acquired a second Revio. So we have 2 Revios, Sequel IIe. They're running 24 hours a day, thanks to Joe's team. But we're also doing a long read sequencing project, also sponsored by PacBio, Google, with the Children's Hospital at Montefiore. This is in the Bronx. 90% of this population is non-white, so we're super excited about that project. But I'll leave it there for John to talk more about that. So our workflow is standard. We get a sample, a DNA sample, we shear it, we do a library prep, we run the samples. And here's a typical sample. If you look here, the mean read length is 16 kilobases. Short read, obviously, we're looking at 120 base pairs for that. So really exciting to have this length. And so we're generating a lot of reads on one sample, almost 8 million. And a lot of gigabases, as you can imagine. Fortunately, the cloud is getting cheaper. When I was at NIH, I remember using the BioWolf computer. I see Alice Young, who I used to bring samples to at NISC when I was at the NIH. Thank you for everything you've done for the rare disease community, Alice. But so this is really cool, having this long of a length. And so the pipeline's new to us. How do you do this? You know, we've been doing short read sequencing commercially for a long time. How do we commercialize? How do we think about a pipeline? And we're using a lot of the PacBio for this project, PacBio tools. And I'm sorry this is, we can give you these slides. I'm sorry this is so small here, but you can see here we're using PBSV, PacBio structural variant caller. We're using deep variant that was developed by Google, also in part of the PacBio pipeline. Also some really cool stuff like tandem repeat genotyping tool we use. I'll talk a little more about that. Paraphase. There's been some stuff in the literature about paraphase. I'll show you some examples of that. So a lot of really cool stuff. We're kind of putting this all together. And then we've got a homebrew system of looking at all this data. We incorporate some of our GeneDx knowledge too into this pipeline. We have this thing called multi-score that it's a machine learning technique where we look at the phenotypes and we try to figure out what genes and kind of incorporate into this really cool pipeline. So anyways, we're working hard. Our team is working hard on this. We call the pipeline Blue Nile. We name everything after rivers, and I feel like every month we have a new version of Blue Nile. We keep on incorporating stuff, perfecting this pipeline, because this is new. You know, this the long read sequencing for us at least is new, and so we've got a great team working on this. So it's going to be a lot of fun. One thing, in addition to the project with Montefiore and Seattle Children's, we're also going back through our own samples. As you can imagine, we have quite a cache of samples that we've called with karyotyping, MLPA, Sanger sequencing, short read, next generation sequencing, and we're using these examples and we're running it through our pipeline just to make sure that we capture everything. And so, and that's another point of this too. We want to make sure that the long read sequencing, captures everything, which it is capturing everything that short read does, and then we want to see what else do we get from it, getting to that concept of a one test. So I'll show you a few cases, and if you have any questions, Joe Devaney's in the back. He can go through these cases, and then I'm going to talk a little bit about the future. And I think I have about 5 minutes to wrap all this up. So there's this concept that's been a thorn in my side for decades: pseudogenes. Pseudogenes really mess things up. These are segmental duplications found in nature, and if you think about this little cartoon here, you've got the gene, the real gene, and then you've got its pseudogene that's been duplicated. Within that pseudogene are some variants that make that gene non-functionable, but when you break up the little pieces, 120 base pairs of short read, and you try to align it to the reference genome, it gets all mixed up here. So you can see the paralog is in red, the pseudogene, and it ends up in the gene. And it really bothers me that we don't call this classic condition that I learned about in medical school called congenital adrenal hyperplasia in short read. This is the most common cause, is a gene called CYP21A2, and it causes genital dysmorphology, salt wasting, and neonatal death potentially. It impairs synthesis of cortisol from cholesterol in the adrenal cortex. But it's very complicated to call this gene, and we don't do it. Most companies don't do it on short read sequencing because there's a pseudogene, CYP21A1P, which is 99% sequence similarity to the real gene, and that really messes things up. And if you look on chromosome 6, here is CYP21A2, and right next to it is the pseudogene, and there's a lot of gene conversion and recombination events during meiosis, when you've got two genes with this similar sequence and close together. So it really kind of messes things up, and that's the first thing I always think of when I think of long read sequencing and short read sequencing. It's like, would love to have congenital adrenal hyperplasia. And there's a lot of other pseudogenes on the NIH website. Here's the reference. There's a long list of these pseudogenes. Many classic syndromes. Another classic syndrome that's difficult to call on short read is Gaucher syndrome, and I had the pleasure of working with one of the top researchers when I was at the NIH for many years, Ellen Sidransky laboratory. And this is really an interesting disease. This is a metabolic disease. It's a lysosomal storage disease. It's caused by biallelic variants in glucose cerebrosidase gene, GBA. It used to be called GBA1. For whatever reason, we call it GBA now. And clinical findings are varied from prenatal lethal to asymptomatic type. There's different types I won't get into. And again, that pseudogene thing coming back to haunt us. This is the long arm of chromosome 1, and you can see GBA is here, and GBAP, the pseudogene, is there. So similar situation. And we looked at a case of GBA with a recombinant allele, and this is the actual report. There's no patient information, just what we found. This is a relatively common scenario. And we ran it through the short read sequencer, and we already knew what the genotype answer was from this case. This was a recombinant event, and we used repeat prime PCR to have this answer here. For the sake of time, I'm just going to show you. I was going to talk about recombination, but just for the sake of time, what we found was there's this thing called a REC-Neal haplotype, and it's on exon intron 9 and exon 10, and it's a gene conversion event. It's very common, and so here's the long read sequencing, and so these two arrows are the pathogenic variants, And this is nice. You run it through long read sequencing, the deep variant piece. This is an IGV view. And here's one allele at the top. Remember I said it's recessive, biallelic, and here's the other one. It's really cool. This is a gene conversion event. This is DNA, and here's the crossover recombinant event with the blue. Let's just say the blue is the real gene, orange is pseudogene. That information is transferred, and here we can diagnose it. We know that this is the two haplotypes. This is not the pseudogene. This is the gene. Here's the variant. Here's the pathogenic variant, the T to C change at position 1448. And this was a gene conversion event that we were able to pick up. Very difficult to pick up on short read when you're chopping everything up into little pieces. So there's Gaucher disease, really cool. I hope you're as excited about this as I am. Repeat disorders, this is really cool. There's all kinds of repeat disorders out there. It's difficult to really get precise diagnoses with short read sequencing. And my favorite repeat condition is FMR1 because I learned about it over 30 years ago when I was in medical school. This is my medical school textbook that I took a picture of. Francis Collins was my professor, and Tom Skelter, and it still has my name up there. I'm an M1, which is first-year medical student at the University of Michigan. And I consulted this book as I was giving this talk, and Fragile X is a condition. It's an unstable CGG repeat in the 5' untranslated region of the FMR1. These are males. They have intellectual disability is the main phenotype, but also some other things, long ears, long face, macrocephaly. Historically, and I had to go back and read, how did we do this back then? Well, it was a karyotype of lymphocyte cultures deprived of folate or thymidine, and there were breaks in bands 27 and 28 on the long arm of chromosome X. And I read my textbook, and it said, we have no idea what gene this is. We don't know what the mutation is, and this is all we know. We get these breaks. Fast forward to right now at GeneDx, we're doing repeat prime PCR, which is, Google it, it's kind of an interesting technique. But certainly, if you think about it, a child who comes to your clinic, has intellectual disability, autism spectrum disorder, you want to look for a lot more stuff than FMR1, and that's what I think the beauty of having a more comprehensive test, as I'll show you. And I'm going to show you some examples of an FMR1. For the sake of time, I'm going to skip to this part. Okay, so this is something called the tandem repeat genotyping tool that PacBio developed. Thank you, PacBio. And this is the tandem repeat viewer. And really cool. These are the repeats here. This is a 29 and 49 repeat. Obviously a female, it's on the X chromosome. And you see blue in red, and that's the methylation. And so if it's red, it's methylated. That's part of the, I'm not going to get into Fragile X, but Google it. If it has more than 200 repeats, it's pathogenic. You get methylation of the CPG bases, and this is really cool. So as you would expect with X inactivation, you should have a 50-50 blue. And I'm going to skip right to the mutation. So remember what I said, mutations over 200. So this is a positive sample, 338. So you can see, and the viewer, you've got repeat expansions here in blue, over 338. And voila, you've got completely methylated allele. So that's really cool. Certainly, if you think about it, I went through that other slide quickly, but with our current assay of repeat prime PCR, we also do a methylation piece on that. This does it all in one step. By the way, our diagnostic rate for FMR1 for intellectual disability at GeneDx, and this is hundreds of thousands of samples, is 0.45%. So you want to look at for this, but you want to look at a lot more things. So that's the nice thing about including FMR1 into a read. And certainly there's I'm gonna skip this, but on the theme of pseudogenes. There's something called incontinentia pigmenti. It's a gene called IKBKG, and there's a classic deletion, exons 4 through 10 that we can pick up. These are some views, IGV views, but this is very difficult to pick up on short read sequencing because, again, it's that thorn in our side, the pseudogenes. So I want to close by talking about future state. And this might be far off, but I think it's a really cool thing that we believe in, that I believe in. We're doing a project called Guardian. Hopefully this publication will come out soon, this summer, our initial publication. And this is newborn screening on a whole genome sequencing backbone. So it's a short read sequencing backbone. And traditionally, newborn screening has really been, for the most part, metabolic diseases that can be picked up on tandem mass spectrometry. And we're interested on turning the system on its head, turning everything upside down, disrupting the way we think about newborn screening, because there's a lot of conditions that are not picked up with tandem mass spectrometry. And this is a cool study. We've gone over 10,000 infants that have been screened for 400 genes on newborn screening. The things we're looking for, 90% penetrance and age of onset before 5. So really interesting project. We've had a lot of changes in management so far of these, some of these kids. One of the most notable is there was a immunodeficiency syndrome we picked up, called the infant back, and very low T cells, and ended up having a bone marrow transplant. So the PI for this project is Wedi Chung. You can go on our website, see all the different genes. So you're probably asking yourself, so what? You're supposed to be talking about long read sequencing today. All right, I'll talk about it in a minute. One thing I will say about the study that blew me away, 75% of parents consented to the study. I was thinking like it'd probably be like 10%. I was surprised. High consent rate. Like I said, we're over 10,000 samples right now. This is the largest newborn screening study using sequencing technology in the world right now. There's a bunch of other projects out there. There's Genomics England that's also doing it. But okay, now let's talk about long read sequencing. Here was, we called this a false positive. Well, we had a number of cases that we were calling for cystic fibrosis on the CFTR gene. We were calling these two variants pathogenic, and we're only doing the proband, right? So we can't phase things, and that's the beautiful thing about long read sequencing is you can phase. I showed you a view of paraphase and IGV views where you can break out the phases. Well, we had to call the parents back, sequence them, and we figured out that these two variants occur naturally, usually in cis, which means they're both on the same the same chromosome, same haplotype. So these kids were also tested with the sweat test, and look, you know, we looked at the phenotype for them. They did not have cystic fibrosis, of course, because both likely pathogenic variants were in cis. So I envision a time, I don't know if I'll be retired or maybe dead by then, where newborn screening is a long read type of event, and especially for phasing is a great example of that. So I'm really looking forward to the future. One last thing about this project, we went back and looked at 25,000 positive, or a fraction of our cases at GeneDx. Almost 100,000 cases that were sent to us by exome sequencing, and based on the gene list we're screening for, we predict that 21% of our cases, our exome cases at GeneDx, would have been diagnosed by Guardian. I know that's a little bit of a leap, and there's a lot of statistical stuff, and the reviewers from our first submission definitely beat us up on this. But we also looked at the average age of these kids. It was 10 years old. And I can tell you there's a lot of stuff, a lot of kids getting diagnosed late that are missing out on intervention. So anyways, newborn screening, the Guardian project, certainly I do see a time when this will be a long read sequencing screen when we get the, based on throughput and price and that types of things. So I'm a little bit over, but James, I think you set me up for being a little bit over. So yes, your fault, James. So I don't know if we have time for questions or.
We can probably take